Hello, everyone. We are going to be looking at some topics in Chapter 10, specifically mitosis, which is a type of cell division. Please make sure that you refer to Chapter 10 in your online textbook as a reference. So let's start out by saying that humans begin life as a fertilized egg that we call a zygote. And then the zygote undergoes billions of cell division to produce a multicellular organism. When a human is fully grown, cell division does not stop. Or we can also say cell reproduction does not stop. Cells need to continue dividing to allow for repair and maintenance of tissue cells. So basically, this is saying that cell division allows growth and development from a zygote to a multicellular individual. And cell division also allows for the maintenance of tissue cells, whether that be repairing the cells or dividing to replace cells that are lost during normal wear and tear. Cells also divide to produce sex cells that we call gametes. Examples of gametes are sperm cells and egg cells. This is going to allow for sexual reproduction and therefore continuance of the survival of the species. Let's also make note that any type of cell division is closely monitored. Later on in the presentation, we will talk about checkpoints during cell division that monitor that cell division is going correctly. In order to talk about cells division, we need to make sure that we understand DNA a little bit better. The entire set of DNA that a cell contains is called its genome. First, we're going to talk about the prokaryotic cell genome very briefly, and then we're going to talk about eukaryotic cell genome, and we're also going to talk about eukaryotic cell division. Remember that our prokaryotic cells are our bacterial cells, and you can learn about more detail about different types of prokaryotic cells in different classes in biology. At this point, you want to make sure that you understand that bacteria is a prokaryotic type of cell. Prokaryotic cells are different than eukaryotic cells because prokaryotic cells are a lot smaller and much less anatomically complex. The DNA inside of a prokaryotic cell, like a bacterial cell, is going to be a loop of DNA, and the DNA is a double-stranded molecule. The loop of DNA seen in a prokaryotic cell, like a bacterial cell, is often coiled and very condensed, and we call this the nucleoid region within that cell. In addition, bacterial cells may have an extra small ring of DNA, which is called a plasmid. The plasmid will end up coding for specialized traits that can be exhibited by bacterial cells. An example of a very specialized trait that a bacterial cell shows would be antibiotic resistance. If you've ever heard about a bacteria that cannot be killed when you take an antibiotic, that would be an example. Let's switch and talk about eukaryotic cells in a little bit more detail. The genome in eukaryotic cells exists as separate individual linear pieces of DNA. So in the prokaryotic type of cell, here's the cell, there is a very large loop of DNA. So that would be the genome in a bacterial or a prokaryotic cell. But in a eukaryotic cell, the DNA is located inside of the nucleus in individual strands that vary in size. So it is not a loop of DNA. It turns out that in a eukaryotic cell, DNA occurs in pairs of linear pieces of DNA, which we're going to call homologous pairs of chromosomes. So when you hear that term chromosome, make sure that you think DNA. And actually, a chromosome is a condensed DNA molecule. Let's show an example of the DNA that we see in eukaryotic type cells. Let's use humans as an example. In humans, body cells are called somatic cells. So body cells, the fancy term for body cells will be somatic cells. And under normal conditions, the somatic body cells have a total of 46 chromosomes. Remember that term gamete means sperm or egg cell. And in humans, the sperm or egg cell will have 23 chromosomes. So I'm going to draw a sperm cell with some DNA inside of it. So remember, we're talking about human cells, and humans fall in the category of a eukaryotic type of organism. So the DNA occurs in pairs. Well, we said that human sperm cells have 23 chromosomes, but I'm not going to draw all 23 chromosomes. We are just trying to understand some concepts. However, you can see my chromosomes are different sizes in length. We can also illustrate that these chromosomes are coded with a different sequences of DNA, and we're going to use different colors to demonstrate that. Let's combine the sperm with the egg gamete. So again, the egg gamete will have some linear pieces of DNA in various sizes. And we'll use different colors to indicate that this is a different DNA sequence. So we have our orange DNA sequence and our pink DNA sequence. They are not identical sequences of DNA. Please do not forget that human gametes contain half of the number of chromosomes as our regular body cells do. We have a term that describes half of the number of chromosomes, and that would be haploid. So anytime you hear the term haploid, just think of it as a cell that has half of the number of chromosomes. So our egg is haploid, and our sperm is haploid as well. When the sperm and the egg combine during fertilization, we make a fertilized egg, which is called a zygote. Now we're going to take the DNA from the sperm and the DNA from the egg and draw all of those DNA molecules in the fertilized egg. Let's do the DNA that the egg inherited from the sperm cell first. You can see the sperm cell had a big purple chromosome and a little green chromosome. And then let's go ahead and color code the other chromosomes that the fertilized egg inherited from the egg cell. When two chromosomes are about the same size and the same shape, we call that pair of chromosomes a homologous pair of chromosomes. So it looks like we have two homologous pairs of chromosomes in our fertilized egg. However, if we were talking about a human, then we would have a total of 46 chromosomes with 23 pairs of homologous chromosomes. I just don't want to draw 46 chromosomes. Each homologous pair is made of a paternal homologue. We call it paternal because it came from the sperm cell. So paternal homologue and a maternal homologue because the yellow chromosome came from the egg. So that's maternal, mom. The cell that is produced after the sperm and the egg combine during fertilization is called a diploid cell. The term diploid can be abbreviated with the symbol 2N, whereas haploid is abbreviated with just an N by itself. Let's go on to the next page. Let's go ahead and define homologous chromosomes. We showed you what it looked like on the last page, but let's go ahead and define it. So, in order to fully understand what homologous chromosomes are, I am going to go back to our original, our drawing of this pair of chromosomes, and I'm just going to talk about it a little bit more. So, that was the pair of chromosomes on the previous page of notes. We have the purple and the yellow. Those two chromosomes are about the same length. And remember that the first one was from the sperm cell, so that's called the paternal homologue. And if you review the notes on the last page, the orange one right here is from the egg cell. So that is the maternal homologue. And together they make up a homologous pair of chromosomes. Each chromosome will have a segment with a specific nucleotide sequence called a gene. So this is a segment of my chromosome, and it's going to be called a gene. On the other homologue, there will also be a gene at the same location on the chromosome, and it's going to end up coding for the same trait. So a gene is a segment of a chromosome that codes for a trait. A very basic example could be that maybe the trait is coding for height, and this gene codes for tall height, and this gene codes for short height, but both genes still code for height. We will learn more about alternative codes seen within genes and discuss the genetics behind that in chapters 13 and 14. Let's review DNA a little bit more. DNA is a very long and very thin molecule. So during certain stages of the DNA, the cell cycle, which we will discuss shortly, DNA needs to be what we call replicate it during a process called DNA replication. And when DNA is condensed, the molecule is referred to as a chromosome. When DNA is not condensed, it is referred to as chromatin. This is the same DNA sequence, meaning it's the same DNA molecule. It could just be a very condensed molecule, or it can be a very unwound and long and thin molecule. We need DNA to be in the form of chromatin, which is unwound and not condensed, in order to allow for a process called DNA replication to occur. We will learn about the details of DNA replication very soon. However, in lab, we will look at cells where we can see chromosomes. When we look at a cell that has its DNA in the form of chromatin, we will not be able to see the DNA with the microscopes that we have in our lab. So let's go ahead and list that chromosomes, which is basically the condensed form of DNA, are going to be visible with our light microscopes in our lab. So the next thing we need to do is talk about the cell cycle. And basically, the cell cycle is just made up of, you start off with one cell, and that cell divides and forms two cells. So we're going to talk about all the activity from the starting cell to the ending cells during this process. Let's go ahead and list the definition of the cell cycle. So the cell cycle is a very ordered series of events involving cell growth and physical cell division that produces two daughter cells. So like I said, your starting cell and the ending cells are going to be called daughter cells. There are two major phases in the cell cycle. The first phase is called interphase. And the second phase is called mitotic phase. And this is going to be the physical cell division that occurs when the cell splits apart into two separated daughter cells. So during interphase, the cell will grow, it will acquire nutrients to support cell division, and most importantly, the DNA will replicate so that there's enough DNA for the daughter cells at the end of the process. Interphase consists of three subphases, G1, S, and G2. During the S subphase, the process of DNA replication occurs. During the mitotic phase, when the cell physically begins to divide and separate, the replicated DNA and also the cytoplasm of the cell will be divided. So the mitotic phase consists of two parts called karyokinesis, which is also simply known as mitosis. And the other portion is called cytokinesis, which is going to be division of the cytoplasm. Moving on to page 3 in our handout. I would like to stop for a moment and talk about a topic that we're going to discuss and look at in lab while we do mitosis and meiosis. We're going to be doing a lab called karyotyping. Let's talk about what a karyotype is. A karyotype is going to be a picture that shows the chromosomes that we've obtained from a cell. So here's the cell right here. Here's the cell, and here's all of the chromosomes that we've obtained from the cell. What we can do is we can use a computer to take all of those chromosomes and organize them into homologous pairs. So a computer will artificially arrange all of these chromosomes into the homologous pairs that we see in this box right here. So this is an example of a human karyotype. And the reason I can tell that it's a human karyotype is because I could count the chromosomes. Looks like there's 2 yellow ones right here. 1, 2, 3, 4. If I count all the chromosomes, I will find out there are 46 total chromosomes, and they're going to be arranged in 23 pairs. A lot of times a karyotype can be developed in order to tell you if an individual has the normal number of chromosomes or an abnormal number of chromosomes. So we know that the normal number of chromosomes in a human is 46. Let's look at the last pair of chromosomes in the karyotype. I'm going to go ahead and highlight them as much as I can against this dark background. So here is the last pair of chromosomes. Right? Pair number 1, pair number 2, pair number 3. I can see all my different pairs. I got pair number 22. Well, this is the very last pair right here at the very end, and I just see an X right there. Make sure that you can locate the last pair of chromosomes shown right there. The last pair of chromosomes are called the sex chromosomes. The sex chromosomes are the genetic designation for female and male. If a karyotype shows two X chromosomes, which this one does, then that is a female. So the genetic designation is female. And if the karyotype is XY, and Y is a very small, tiny chromosome. So if you see an X and it's paired up with a little teeny tiny chromosome next to it, that would be a Y chromosome, then the genetic designation is male. So in this example, it is going to be a female karyotype. All right, let's complete the rest of the work on the page. The term aneuploidy refers to a condition of a cell having either an extra chromosome or a missing chromosome. So types of aneuploidy are going to be monosomy aneuploid. That's going to be an individual that's missing a chromosome. In humans, for example, that will be when the individual has the diploid number, remember that's diploid, -1. Okay, so diploid is 46 -1. That means this individual has a total of 45 chromosomes, which is not the normal of chromosomes for humans. So one example of that is a female with only one X chromosome. Sometimes the genetic designation is written like this, only indicating only one X chromosome. So that individual will have a total of 45 chromosomes. We'll mark off this other example. The other type of aneuploidy is called trisomy, and there are more examples of this one. This is going to be when an individual has an X chromosome. So in humans, this is going to be the diploid number +1. That equals 46, because the diploid number is 46 in humans, +1. That means these individuals have a total of 47 chromosomes. So an example of that is something called trisomy 21, which that individual will have 3 of these 21 chromosomes right here. See the number 21? They'll have 3 chromosomes at the site of 21. Therefore, they're going to have a total of 47 chromosomes. This is also called Down syndrome. This can occur not only at chromosome 21, but other chromosomes in the karyotype as well. Another example would be an individual who has 2 X's plus a Y chromosome. This is going to be a male with an extra chromosome. Another example will be XYY. This is going to be a male with an extra Y chromosome. So there are a lot of aneuploidies that are viable when we talk about sex chromosomes specifically. There is another example of this, and you're going to determine that in lab. Okay, we want to get back to the cell cycle. Okay, I want you to remember that the cell cycle is when you start off with one cell and it divides and it produces 2 individual cells. So it's going to be all of the activities from the starting cell to the ending cells. You can review figure 10.6 in your textbook. Let's go ahead and expand that picture. I am not the best at drawing, but we will expand that picture, that up here, and we're going to add some details. So the cell cycle is going to be all of the activities from the initial parent cell. This is our starting cell. You can sometimes think of this as a stem cell. To the cells produced at the end of the process, which we're going to call daughter cells. And there's 2 of them produced. The first portion of the cell cycle, remember, it's made of 2 parts: interphase and mitotic phase, which we learned about on previous pages. So the first part is going to be interphase. And this lasts from 70 to 90% of the cell cycle. So it takes a very large chunk of the cell cycle. And then the second part of the cell cycle is going to be called mitotic phase. So let's go ahead and recall that interphase is made of 3 subphases. If you need a moment to look back, then please look back in our notes. But the first portion of interphase is called G1. Okay, G1 consists of when the cell is growing and acquiring nutrients. The cell will then continue into the next subphase called S. And the cell will continue into the next subphase called G2, where the cell will continue growing and checking DNA for accidents. Okay, when G2 is completed, the cell will move into the mitotic phase, which is the physical cell division. That's when the cell begins to prepare for splitting apart to produce 2 separated daughter cells. So you can see this is definitely a sequence of the events that are continuous, more or less. Let's go ahead and highlight the S subphase. We want to really talk about this S subphase. And this is what you want to know about the S subphase. During the S subphase, DNA is replicated. Let's just take a very basic and brief look at what that looks like. So the parent cell starts off with a piece of DNA. We call this independent DNA. And then during the S subphase, a process called DNA replication occurs. And now we have 2 identical DNA molecules that are more or less connected together. This DNA is now called a duplicated DNA molecule because DNA replication occurs and you have 2 identical DNA molecules that are ultimately held together at this point. These 2 identical DNA molecules are called sister chromatids. And they're held together in a region that we're going to call the centromere. So the centromere region is keeping the sister chromatids stuck together. Now let's go back up to G2. So now that we have undergone DNA replication, okay, the cell continues on to the next subphase, which is G2, which is another basically checkpoint during the cell cycle where we check out the DNA, make sure DNA replication occurred correctly before we proceed onto mitotic phase. The mitotic phase is made of a set of subphases that will physically divide the cell up into two daughter cells. Those subphases are called prophase. This is prometa, anaphase, metaphase, anaphase, and telophase / cytokinesis. We're going to talk about these in more detail on the next page. One thing that we want to know right off the bat is that during the mitotic phase, these two sister chromatids are definitely going to be split up, and each of the chromatids are going to end up in each of the daughter cells. However, when the sister chromatids are split apart during mitotic phase, they're going to then be called daughter chromes. Excuse me. That's called daughter chromosomes. So let's go ahead at this point, even though we have not talked about the specific stages of the mitotic phase, let's talk about the overall order of cell cycle going through cell division. In this case, the cell division type is called mitosis. So we start off with G1. We continue on to S, and that continues on to G2. After that stage is completed, we move on to a stage called prophase. Then we move on to a stage called prometaphase. I'm going to put it in parentheses because that stage is sometimes not added in your lab manual. Then we will move on to metaphase, anaphase, telophase, and cytokinesis happens at the same time. So remember, these three events occur during interphase, and then the rest of these events occur during the mitotic phase. On the next page, we are going to be talking about these events right here. Please go ahead and pause the video so that you can copy down the basic drawing if you would like. I am going to proceed through the stages of the mitotic phase. You can refer to figure 10.6 in your online textbook. It's pretty similar. We're going to use an animal cell as an example. So this could be humans. The first stage that we're going to start off with, so we understand some things, is G2 of interphase. So here is a cell that's in G2 of interphase. Let's talk about a few things within this cell. First of all, this is the parent cell. And remember, if we're talking about an animal cell, which is eukaryotic, like a human cell, then the cell is going to have that big round ball structure, which is called a nucleus. And the DNA is inside of the nucleus. And during interphase, the DNA occurs as unwound and not condensed. So the DNA is in the form of chromatin. When DNA is in the form of chromatin, this allows the DNA to be replicated. Okay, so remember the DNA was replicated back in the S subphase before G2, but DNA is still in the form of chromatin at this point. There is also a very light, round little dot-like structure, okay, within the nucleus that is going to be called the nucleolus. Please do not forget that the nucleus is surrounded by that gooey fluid that we're going to call the cytoplasm. These little bar-like structures over here that have some little threads growing out of them are called centrioles, and those little fibers growing out of them are called spindle fibers. So we're going to see that they play a role in moving structures within the cell. So when the cell proceeds from G2 of interphase to prophase, now the cell is in mitotic phase. It is preparing for physical cell division. The mitotic spindle, which is basically these little protein fibers, begin to develop a little bit more. At the same time, the nuclear membrane, the membrane of the nucleus, begins to fragment and disappear. In addition, we no longer see the nucleus during prophase. The nucleolus has disappeared as well. We are now starting to see some DNA forming in the cell, meaning that we were not able to see the DNA in interphase. We're able to clearly see a nucleus and a nucleolus, but we, with a microscope, cannot see the DNA. But now that we move to prophase, the chromosomes, which is the DNA inside of the cell, are visible when we use a microscope to view the cell. So what I would like you to do is make sure that you know some events of each stage that indicates what stage that we are in. Basically, for prophase, some indicators would be chromosomes become visible. The nuclear membrane is seen fragmenting. We can no longer see the nucleolus during prophase. Our next stage is called prometaphase, and I'm not sure that it's going to be covered in portions of your lab. It's not in your lab manual, but let's go over it very quickly because This is a continuous series of events. During prometaphase, the chromosomes, and the chromosomes are going to be shown in pink right here, just for the ease of drawing the chromosomes. The chromosomes attach to what we call the spindle microtubules. This is all a type of protein that is growing out of the mitotic spindle. That word is microtubules. And it's basically a type of protein which will connect to the chromosomes in order to organize the chromosomes, to prepare the chromosomes to be pulled apart during physical cell division. Our next stage after prometaphase occurs will be metaphase. So we're going to continue on the bottom column here, or the bottom row. During metaphase, the chromosomes are all lined up in the center of the cell. So the microtubules have organized the chromosomes at the center of the cell, and the center of the cell is called the metaphase plate. If we want to make sure to evenly divide the DNA up, we have to put all of it at the center so that all the DNA can be pulled apart evenly. And that is the function of metaphase. So the way I remember it is metaphase, middle. Chromosomes are lined up at the middle of the cell. The next stage is going to be called anaphase. And this is going to be when the daughter chromosomes are pulled apart to the opposite ends of the cell by the microtubule fibers. So the daughter chromosomes are pulled apart to the opposite poles of the cell, kind of like the North Pole and the South Pole, because eventually what we want to do is evenly separate the chromosomes so that each daughter cell, and this is going to be the first and second daughter cells, those are going to be the two daughter cells right there, are going to have the exact same number of chromosomes. So in a human, that would be 23 chromosomes in one daughter cell and 23 chromosomes in another daughter cell. I'm sorry. That's for meiosis. Please strike that from the record. Okay. It should be 46 when we're talking about mitosis. Okay, so these two cells, which you can see are being separated as that little indentation grows farther and farther to eventually completely separate the cytoplasm, okay, is going to be, these two cells are going to be the daughter cells produced at the end. So this is what we call telophase and cytokinesis. Telophase is when two nuclei are formed. So two nuclei are formed, and you're also able to see the nucleolus. So when two nuclei are formed, that is telophase. We can also see the cytoplasm is dividing in half, and that is cytokinesis. So the cytoplasm dividing in half by forming this little indentation. Okay, let's talk about cytokinesis a little bit more. Try to organize it so it's, there we go. Let's talk about cytokinesis a little bit more because it happens differently in animal and plant cells. Okay, so in animal cells, cytokinesis is accomplished by forming an indentation called a cleavage furrow to separate the cytoplasm. So it kind of looks like this, just like the picture up above, like a figure 8. Here's the 2 nuclei and the 2 nucleolus. Okay, here's that indentation, which is a fissure, okay, formed in the cell by contractile proteins. Kind of like how your muscles have contractile proteins to allow for you to flex and extend your muscles. There are contractile proteins that squeeze and squeeze and squeeze and squeeze until these two cells are entirely pinched apart. So we call that indentation a fissure. That fissure is called a cleavage furrow. And that indicates that cytokinesis is occurring. Furthermore, it is a great indicator that the cell is in the stage of telophase. So what's going to happen is that we will have 2 separated daughter cells at the very end of the animal cytokinesis. Plant cells are confined by their cell walls, so they undergo cytokinesis a little bit differently. Plant cells form a structure called a cell plate to separate the cytoplasm to produce 2 individual daughter cells. So here's a plant cell, and plant cells form these stacks, which are called, they're formed by the Golgi apparatus, and they're basically filled with sugar. Okay, so it looks like a little line it formed in the middle of the cell. This is called a cell plate. And what's going to happen is that this will continue until the 2 cells are completely separated. Two separated daughter cells. So the major difference between a plant cell is the plant cell makes a cell plate to effect cytokinesis, and an animal cell makes something called a cleavage furrow, which is an indentation of the cell in order to effect cytokinesis. Okay, so let's just quickly talk about some more ideas when we're talking about cell division. Do all cells and tissues in a eukaryotic organism, for example, humans, go through the steps of the cell cycle throughout the organism's life? No. Okay. So there are some cells which will not continue to undergo mitosis or cell division throughout your entire life. But a lot of cells do continue to divide throughout your life. So, for example, cells that are very easily damaged, even through normal wear and tear, tend to have a lot of cell division going on. So, examples of cells that are very easily damaged just by normal functions are going to be digestive cells, skin cells, and blood cells. Some cells do not continue to go through cell division. Instead, they stop at a stage called G0 in cell division. So, two examples of cells that basically do not continue cell division, instead, they take a detour into cell specialization, will be neurons. That's going to be cells of the brain, spinal cord, and nerves, and cardiac muscle cells of the heart. So, because these type of cells do not continue to undergo cell division, we do not want to damage them. In fact, a lot of times we build structures around them. So, for example, your brain has bone surrounding it. It also has meninges, cerebrospinal fluid. There are basically layers of protection that make a huge effort to protect these cells so we don't damage them, because if these cells are damaged, they cannot regrow from cell division. It just takes an incredibly long time to grow these cells and get them very highly specialized for their function. So you want to make sure that you understand not all cells are able to continue through cell division. Let's just very quickly talk about, do cells in a eukaryotic organism undergo cell division at the same rate? No, certain cells undergo really high rates of cell division. Some cells last a little bit longer, so they don't have to undergo as high of a rate of cell division. This really technically is determined by how long the cells last. So, for example, digestive and skin cells, remember, digestive was your stomach and intestinal, they basically don't last very long. So there is a very high rate of cell division to replace these cells. So they may last approximately 2 to 5 days. There's a high rate of cell division in these tissues in order to replace cells that are damaged during normal function. Blood cells actually last a little bit longer, even though there's a lot of cell division going on in the marrow. Blood cells, a typical blood cell may last approximately 120 days. So, just different cells have different lifespans, and different tissues have different rates of cell division. Unfortunately, cancerous cells divide at very high rates. So, they proliferate a lot faster than other cells in the body. They basically are the effect of uncontrolled cell division. How do we target these type of cells to make an effort to destroy cancerous cells without as much damage to the other cells in the body? That's what chemotherapy tries to do. So chemotherapy really targets quickly dividing cells. Unfortunately, you do have other cells in your body that divide at very high rates, and they could also end up being targeted by chemotherapeutic drugs. So a lot of times, especially skin and digestive cells, um, because of their high rate of cell division, we're limited by how concentrated that chemotherapy can be, because otherwise we're going to really not only damage and kill off the cancer cells, but we're going to destroy also our very fast dividing cells as well. All right, let's talk about the control of the cell cycle. So we don't want to let the cell cycle go uncontrollably. We have to be able to control the progress of the cell cycle so we don't have tons and tons of cells. So the cell cycle is controlled both by external and internal mechanisms. I'll make sure it's clear. We're on page 7. So examples of external control, okay, that could either inhibit or initiate cell division. So inhibit is stop cell division, and initiate is, hey, let's let cell division go. An example would be if human growth hormone is around the cell. So if the cell is receptive to human growth hormone and there's human growth hormone around the cell, that will initiate cell division. On the contrary, if there is a lack of human growth hormone, then cell division will not progress. Another example of an external event that will affect the rate of cell division, it will be cell crowding. This means that many cells begin to crowd a space, and this triggers the cell to stop going through cell division so much in order to control the amount of cells in a tissue. So let's go ahead and quickly discuss the internal checkpoints. They're going to be located at G1, G2, and M, which is basically going to be around metaphase. And you can review section 10.3 in your online textbook. But what I wanted you to understand is that we do have checkpoints during the cell cycle in order to regulate cell division. And if the cell senses any sort of damage or abnormality along the way, the cell attempts to either repair or just kill off the cell to prevent it from proliferating. So a few regulator molecules that control the progress of a cell cycle. First, we have our positive regulators. They're going to promote cell division. So examples of these molecules are called cyclins and cyclin-dependent kinases. Basically, the presence or the activated presence of these substances in the cell will increase cell division. These are intracellular molecules, and increases in the concentration of these molecules are going to be triggered by external and internal signals. Something to note about these regulator molecules. They include a complex series of reactions that promote cell division. And I don't expect you to know the series of reactions, but you should know it is a series of chemical reactions that produce a product at the end, which will then promote cell division. So that means that not just one reaction can allow for cell division to continue. It's the reason it's complex is to exhibit some control over cell division. This is a complex series of reactions that promote cell division. Okay, we also have negative regulators that inhibit, they stop cell division. So examples of these are simply called P53 and P21, and this is just done on some research on the molecule, very basic names. Okay, what these molecules ultimately do is they block the cell cycle from continuing. They're going to go ahead and do this if they detect that DNA is damaged um, at any time during the cell cycle. Another example of a molecule that is a negative regulator, it has a really long name. It's called retinoblastoma protein. It's abbreviated R(b), and it's found in a group of molecules called tumor suppressor proteins. Ultimately, they're going to block molecules that are needed to progress cell division. All right, let's wrap up this section and just go ahead and note that in cases of cancerous cells, these regulatory proteins were found to either be damaged or non-functional. They play such an important role in controlling cell division, not allowing cell division to occur if there's any abnormalities or damaged DNA, that if these regulatory proteins themselves are damaged or non-functional, then that's going to end up resulting in uncontrolled cell division. We want to go ahead and note that if we can repair any abnormalities or damaged DNA along the way, we will attempt to repair it. So if we cannot repair, then cells may undergo a programmed cell death called apoptosis, which basically just when the cell commits suicide and kills itself so it can no longer divide and proliferate its abnormalities. Please make sure you see on 12, question number 12, you have a little on your own activity. For the last page of the presentation on page 8, how do prokaryotes like bacteria undergo cell division? They go through a process called binary fission. They do not go through mitosis. The reason bacterial cells cannot go through mitosis is because they do not have a nucleus. Only cells that have a nucleus are able to go through mitosis, which we call mitotic cell division. That's what we just studied on the previous pages. Thank you. See you at the next lecture.